remember, the next test will be on the Unit 3. It'll be over these 3 chapters right here. And as I told you last time, there's not a lot on the one we were talking about, the cell membrane. So, we were just getting into these things on water balance. I want to run through that real quickly with you and make sure you understand what it is that you need to get from there. We had talked about the basic structure, and now we were talking about how things go across the membrane. And this can be a little bit confusing from time to time. So, try to keep up here. We talked about osmosis and how things went across the membrane. There are proteins, remember, and you don't have to remember everything they have got written down here. Just remember, a transport protein is going to move things across the membrane. That's what they do. They are a carrier. They can move things across. Aquaporins are the particular proteins that move water. And if you're wondering about how they do it, if you notice it says these proteins can bind to the molecule and change the shape that will allow things to cross the membrane. Think of it this way. This is not exactly what happens. Let's say we got a molecule that's that shape, okay? We've got a channel in the membrane right in through here. Another protein could come along and attach to this and change the shape of that protein so it will fit. Now that's an exaggeration, but it explains exactly what's happening. That protein will actually change the shape of that molecule so it can match up to the receptor and go down through. And that's why many of these can't get through until that occurs. And that's a really blatant exaggeration right there, but it does show the basic thing. Know the definition of diffusion, and that's for molecules, the tendency to spread out evenly. Whenever they reach equality on both sides, like if we had a U-shaped tube, a membrane in the middle, we put water on one side, that water will diffuse through that membrane until the water levels are equal on both sides of that tube. That's just plain diffusion, nothing else affecting it. And that shows you the example till we get over here on the end where we get the same number of solids or molecules. Understand what a concentration gradient is. On most things, there is going to be a certain concentration for things to happen. That spread out division of these molecules is called the concentration gradient, like a scale. And it says the concentration gradient is the region along which the density of a chemical substance increases or decreases. That's its concentration gradient. The concentration gradient can go up and increase, or it can go down and decrease. No work must be done to move the substances down this gradient. There is no energy being added to it. It is just what is there naturally. The diffusion of a substance across a biological membrane is called passive transport. Key to it, no energy is added to make that happen. Osmosis is the term specifically used to describe the diffusion of water across a membrane. Notice here, this is very important, water diffuses across a membrane from the region of a lower solute to the region of a higher solute. And some of these things can be difficult sometimes to keep them straight. But if we have a cell that has a low concentration of molecules, whatever they are, and then it is into a solution surrounding it that has a high concentration, has a lot more of those molecules, one of the things we might think is, well, it should go from, all this water should go from the high. It don't do that. Nature says, and this science, the physics, when water is in, or a solute is in the low concentration, like right here, it will send water to this high concentration because it wants to dilute that high concentration down to where it's at. It wants to dilute it down. This cell loses water. This shows just a membrane where that's happening. If we had sugar in the water, it's eventually going to even out with osmosis. These are the ones to make sure you understand, and you pretty much be guaranteed you're going to see those on the exam. Tonicity is the ability of the surrounding solution to cause a cell to gain or lose water. In an isotonic cell, the concentration is equal inside and outside of the cell. Remember, whenever I say outside of the cell, it's talking about just the extra cellular fluid. If the solute is greater than outside the cell than it is in, like the situation we've got over here I drew on the board, that is said to be hypertonic. It's all referring to the concentration outside the cell. If the concentration outside is greater, it's hyper. If it's lower outside the cell, it's hypo. Now notice here, in a hypertonic solution, the cell loses water. Why? Because the concentration is greater outside of the cell and it's trying to dilute it. Hypotonic solutions, the concentration is less on the outside. So, since the concentration of the solute is greater inside of the cell, it will send water into that cell to try to dilute. And we get this situation that we looked at with the red blood cells. In a hypotonic solution, concentration is greater inside of the cell, so water enters the cell and eventually causes it to rupture, just to burst, to explode. In an isotonic solution, nothing happens. That's normal. That's where we want to be. In a hypertonic solution, in which the concentration is greater outside of the cell, then the cell loses water. Where this one exploded, this one implodes. It just folds in on itself.
Why does it say normal for the hypotonic one?
Because that's what we want in our body. We want it to be equal so there's none coming in or going out.
Well, no, I get it for the isotonic, but I meant for the cell wall image.
Oh, down here you mean?
Yeah. Why
That is referring to a plant cell.
okay.
And the reason it's saying that, in a hypotonic solution, remember, there's a lot of water that comes in. Plant cells will have mechanisms to control that.
Oh, okay.
And they actually need more coming in. They can control it. They have vacuoles and things, openings. They've got actually water channels they can open up.
Yeah.
Water will never get too much. And keep in mind, there are some plants that actually live in water. You have to have the method of controlling it. The problem comes when it's too dry. When the outside environment is too dry, they start to lose water to it.
Yep.
And that's what causes them to wilt. Osmoregulation. The control of solute concentrations and water balance is necessary adaptation for life in such environments, what we were talking about. They have the ability to do that osmoregulation. Just talking about the little protostare parameciums. I've been studying those under a microscope before in the past, and the light would start to dry out the water on the slide, and I've actually seen little paramecium going along, and then suddenly just pop, they were gone. You saw nothing. They were just there and poof. And that's because they imploded. They were losing all their water to the outside environment, and they were gone. Many animals have adapted. Let me give you an example with fish, which is very good. Hopefully everybody realizes we've got saltwater fish and freshwater fish. So the solute we're talking about now is salt, right? Even freshwater fish need a certain amount of salt, sodium, the electrolytes in our bodies, we need a certain amount. So because fish that live in fresh water don't get very much salt, they have an osmoregulation device, the kidneys. Those kidneys are designed to conserve the salt and get rid of the extra water, but to keep that solute because it doesn't have enough in water. So what happens if you put a fish, or if you take a saltwater fish, it's designed the opposite. It's living in such a heavy concentration of salt, it needs to get rid of a lot of it. So its kidneys are designed to get rid of almost all of the salt. So if you take a freshwater fish and put it in salt water, the freshwater fish's kidneys are designed to conserve salt, so it dies. I mean, it's just got such an overload of salt in the system, it kills it. Fresh or saltwater fish the same. Its kidneys are designed to get rid of almost all of it. You put it in salt water, now it can't function because it's getting rid of all the salt. There are some fish who live in brackish water. They can go either in the ocean water or they can go in fresh water and survive. Salmon. Salmon live all their life in the ocean, normally in salt water, and whenever they get a couple years old, 2 or 3, they then go up freshwater to lay eggs. So they can go either way with that. They can regulate it. But that is an example of how we must regulate the solids. What do we do? One way of curing meat is to put salt on it, right? What does it do? It draws every bit of the water out of those cells. If there's no water in the cells, enzymes are not going to work that cause decay, and bacteria can't survive that cause decay. So we are preserving it simply by removing the water from it by adding salt. In some restaurants, they actually cook fish with salt. They will take a big fish, clean him up, and just pack inside and on the top in just a big cake of salt and leave it that way for a day, and you come back, basically the fish is not cooked, but it's had all the water taken out. There's not going to be any parasites in it or whatever. Fish are something I have a hard time. I mean, I've seen so much about fish are nasty creatures. I had one guy who was in class like this probably been about 5 or 6 years ago. And he worked in one of the big stores, I won't say which one, but his job was to pull the worms out of the fresh fish fillets they brought into the store. And if you've ever seen a fish fillet and see that little hole and the little brown spot around it, that's where a worm was. And guys like him pull them out. Now, animals have parasites. Pork is real bad for it. Fish is real bad for it. But if you take the moisture out and you cure pork or you cure the fish with that salt, it's safe. There's nothing to it. So I'm not a big fish eater anyway, but man, I've seen some ugly worms come out of them.
I'm sorry, what kind of fish fillet are you talking about? Are you talking about like the Alaskan salmon, just fish, just raw?
Doesn't matter, any fish that's got the big fillets, doesn't matter.
Just raw, that one they just, wow.
Yep.
So osmoregulation in fish, the liver controls the concentration.
Kidneys.
Kidneys control the concentration. Is it in the bloodstream or
Yeah.
how does it?
What happens is the blood goes in through the kidneys and the
Yeah.
kidneys then filter it out.
Okay.
So it goes back and then the blood is whatever the kidneys put out.
Okay, that makes sense.
So the freshwater fish's kidneys are adapted to get to get salt, but the,
No,
oh,
the freshwater fish you put them in salt water will die because they can't adapt. There are some fish that can live in either because their kidneys can adjust.
okay,
That's like the salmon.
okay. But, so the freshwater fish, because they would absorb too much salt, so they wouldn't be able to survive.
Their kidneys are designed to conserve, not to get rid of.
Oh, okay.
And guys, about parasites, this is a true, actual story. And if this doesn't make you want to eat raw fish, I don't know what will. There was a Swedish sea captain, and they would go out on the boat, they were fishermen, and he would always eat some of the raw fish. And this was documented. The guy passed a 32-foot long tapeworm where he had gotten it from eating raw fish. Tapeworms will just fold over like a ribbon in your intestines and stuff. That's a big worm, folks. Telling you. And
Oh my.
eating raw food such as fish, pork, and most animals, wild game animal, is not a good idea at all. Unless, now, a lot of people like sushi, it's fine because it's treated with either an acid or salt. That's good. There's nothing wrong with that. But you have to, heat can kill the parasites, the eggs that are in that meat, or acids or salt can do it too by dehydration. So it's safe as long as you treat it like that. I don't want anybody like sushi, I don't want to make them think it's bad, it's not. There is another animal that can regulate how much water. Notice that little thing, the contractile vacuole? That's paramecia, and it can open and close that and either let water in or out, so it can live in different pressures. We've already basically looked at that. In a plant cell, in a hypotonic solution, it becomes firm because it's got a lot of water coming in. Flaccid is when it starts to wilt. It loses water. And here's the bad thing, as we mentioned. If a plant loses water and the membrane pulls away from the cell wall because it doesn't have enough tonicity, that is called plasmolysis, and it will kill the plant. When the membrane pulls away from it, when it gets that far, then it's not going to recover. Sometimes the plant can start to wilt, you can add water and it'll pop back up almost in front of your eyes. Membrane is not pulled apart. So different plants can go to different extents. Facilitated diffusion, that's where proteins help the process. It gives examples, don't remember those yet. We will see stuff again. Notice up here, we've got a gate. Notice down here that gate can open and shut. So it can open up and let an ion or molecule in, close at the end, and then release it. That's the way many of these work. Now, not wanting you to try to remember the names and all of them. Just, facilitated means there's a molecule that helps it in. Yeah.
Oh, I was going to ask, so would this be considered so this is active transport? Active transport?
Can be. Usually it is because of energy. Anything that takes energy is what's active.
And then that would be considered passive transport because it just flows.
Not one.
Yes.
Channel protein, yeah. As long as it's not doing anything except just providing the way, but nothing's helping it. In other words, get there.
Okay, thank you.
Notice some diseases are caused by malfunctions in specific transport systems. Kidney disease is one. Only thing I want you to know about active transport, it requires energy. Active transport requires energy. One type of active transport that I do want you to remember that you will see here in other situations is called the sodium-potassium pump. This happens on membranes to increase the concentration gradient of the membrane. Now, what'll happen with membranes many times is we need a concentration to get communication, like we build up, just think of it like it's a spark going across that membrane to send a message. That's what happens with sodium and potassium. These are proteins that can take sodium, as you see, and bump them outside of the cell, and it can take potassium and bump inside of the cell. Now, remember when we talked about the electronegativity of atoms? This is not the same thing, but think of it. Some atoms just have more of a charge than others do. So if we take those things with a high positive charge put on one side and those with a low on the other, now we've got a concentration gap that can jump across. When you take cell biology, that will be explained much better for you. So this shows passive transport and then active. Remember the difference between passive and active. Active requires ATP. This shows you the membrane potential I was talking about, is the voltage difference across the membrane. Voltage is created by differences in the distribution of positive and negative ions on the membrane. Two combined forces collectively called the electrochemical gradient. Remember, chemicals, these elements, atoms, have charges. They can make a gradient. Don't worry about these electrogenic pumps and proton pumps and things like that. You will get into those things in cell biology. Right now, just notice, this is one I do want you to remember. When we get into the next chapter, we will see at the end of us eating food, it's all going to come down to taking the chemicals from that food, removing the hydrogen, and pumping them across the membrane of the mitochondria. That is done by a proton type pump. And notice what it does. If we take positives and put them on one side of the membrane, and we've got all these negative charges on the end, what have we got? Positive and negative gap. What happens to that? Positive and negative attract. When it gets strong enough, it'll go across the membrane. It will get strong enough so the membrane can no longer block it and come across. If you guys, none of you are old enough to know anything about the world, I don't think. I can actually remember folks, the first time they brought a computer in my classroom, and I almost fought with them. I told them, you get it blanked out, no one would ever use them. They would never go anywhere. I was wrong, quite often. Cell phones. I remember whenever, not only did you not have a cell phone, but you had a phone in your house. It was, you may have 8 or 9 other families on the same line, party lines. Another thing you guys don't remember is TVs that actually had a picture tube probably in them. Had the big tubes in the back. When you turn one on, it could take up to 3 minutes for it to get enough charge in it to actually, the screen to lighten up. And they had in them capacitors. And a capacitor is an electrical device that does this right here. It's got plates and oil in it that's separated. It has, when you put electricity to it, you'd have a negative charge on one, a positive on the other. So then, when you turn your TV on now, what happens? It's on, because it's got a reserve charge built into it. And if you ever notice on the back of TVs, particularly those old tube ones, it says, do not take the back off. There's a shock hazard. And there has been more than one person killed because they went into the back of the TV trying to get parts out and take a screwdriver and cross them over the positive and negative terminals of a capacitor that was in there and has more than enough electricity to kill you. And that was the way the old tubes were, TVs. You don't see that now. Different technology. TVs come on whenever you hit the button, pretty much. So there's a lot of things that's changed, and I'm not talking nation history. I saw my first computer they tried to bring into the room in about 1993. It's not been forever, folks. Custer's been dead 100 years. guys know who Custer was?
General Custer?
Yeah. On Wednesday, we will begin with the next chapter, 8, Energy.